What's up, Bio Penguins? So we are coming to you live for our Q&A over Unit 4 and 5. We're going to do a little bit of 4 and a little bit of 5. Hey, APsolute RecAP. And Melanie should be joining me in just a moment so that she can join in, go live. Change my camera so that I'm no longer sharing this picture. Stop sharing. All right. So she should be able to get in now with no problem. Wait, where she goes?
I'm right here.
Okay, awesome.
Hi. I know it bumps off the graphic. I learned that the other day.
No problem. I put the graphic up for just that quick moment so that I would have it. And then I forgot because I didn't expect you to get on so fast. So welcome everyone.
Nappy.
So before we get too far in, because we always seem to forget this, can we introduce ourselves?
Absolutely. Hello, penguins and recappers. My name is Melanie Kingett. I am the founder of the APsolute RecAP, and I am the host of the APsolute RecAP Biology Edition podcast.
And I am Tiffany Jones from AP Bio Penguins, and as John from Marker Learning always says, I'm the CEO of this single account of me running it by myself, but he still calls me a CEO. And I go, yeah, sure, I'll just take that title because that's just a lot of fun.
Isn't it funny how much people assume there's like a team behind what we're doing?
No, it's just me, all me. Sleepless me. So hey, real quick before we get started.
Yeah.
Do you know what today is? Like, what's today's date?
It's 3/21. I do know what today is.
Yes. So in the chat, if you can tell us what does it mean by it being 3/21? What do you think it's
heredity.
National? Yeah, think about it. It's a
3/21.
national day for something. Thinking about that, it's March 21st, 3/21. Anyone in the chat? I even have a shirt to kind of like symbolize it. A T21. I think it's backwards to the camera. Sorry. But it's for T21. I'm not seeing anybody chiming in. But it is now. It's National Women's Day month, I think.
March 8th, I think, was National Women's Day. Yeah.
So today is National Down Syndrome Awareness Day. So trisomy 21 having 3 sets of chromosome 21. So I figured I would wear my my trisomy 21 shirt just to support the occasion.
Let's just pretend that we planned it this way to cover
totally
chromosomal inheritance on the day.
We totally planned it.
Yeah.
I made the calendar to specifically do this. So tonight we're looking at Unit 4 and Unit 5 and a little portion of 5. And 5 is going to overlap between this session and the next session just because we didn't have enough weekends. So we just figured that. Plus, I don't know how your teachers do it, and I don't know how you do it, Melanie. But to me, it's logical to teach mitosis and meiosis together.
I always do it together. Yeah.
But the CED actually has them separated with 4 is in, I'm sorry, mitosis is in Unit 4 and meiosis is in Unit 5. And so, like, I figured that we would at least have those together.
Yeah, I mean, we'll definitely end up overlapping some topics. If you've been in our lives before, much of our review is question driven. So if you do have questions, pop it into that question box at the bottom and we'll try to get to those as we do for units 4 and 5.
And just so we're clear, the question that she's talking about, it looks like a little question mark. Looks like a little like thought bubble with a little question mark. If you type it in the chat, that's fine, but we can't see it. Because I literally see one person at a time because I see Melanie's face.
Yeah, and it's partially covering my face, so it's
it will, it's going to partially cover your face once I start looking at these questions.
It's okay.
So we have
just curious, Tiffany, before we start, where are you like in your content right now? I know everybody, teachers are, this is about the time when the anxiety creeps in, but there's a lot of variation with where people are in the curriculum. So where are you?
I'm done.
Yo, your students are so lucky.
So technically my students are done. We finished ecology 2 weeks ago, or no, over a week ago. And we're now doing what I call hodgepodge. So I'm gonna do a little bit of plants and I'll do a lot of animal systems, and I'm gonna connect the animal systems to the different functions. So when we look at, I don't know, the kidney, we're looking at diffusion.
Right.
When we look at, the circulatory system tomorrow, I'm not really looking at anything but gas exchange in the capillaries. So like I look at animal systems, I'm kind of honing in a little bit and trying to get application at.
More exposure,
where are you at?
More exposure to examples. That's definitely ideal. I am in the middle of ecology, so I do go in order. of the CED, and we started a little bit later this year. So I'm about like a week behind where I wanted to be. I will wrap up ecology next week, but I start my review. Actually started my review as early as November. So I do like Throwback Thursday, and so every Thursday I pull something from the previous unit. Sometimes it's like a grab bag. Sometimes it's just the previous unit. But you know, all that being considered, personally, I think units 1 through 6 are the hardest. 7 and 8 are the easiest from like a conceptual standpoint because you get to zoom out. So if you are still in those earlier units, I wouldn't freak out yet. There's still a lot of time to kind of get the global picture.
And Samantha just wrote in the chat that she's in unit 5, which is perfectly fine. Just remember, you don't have to make 100 on the exam, right?
Right.
So if you get through as much as you can get through, okay, and you have a good foundation, a good application, and you know that stuff really well, you can apply that stuff, okay? And anything that you haven't learned, just use your science practices, think through things logically, conceptually, and you could probably figure out some of those
answers. It's like
answers.
Jeopardy, right? Jeopardy contestants don't know everything. Jeopardy contestants can interpret the questions, right? They find the clues in the questions.
And a lot of the times, especially with free responses, you can find points even without knowing some of the biology. Especially now with the way that this formulaic way they're writing the free responses, like you can look at it and know exactly where they're going to ask you the content questions. The content question is always going to be part A. It's going to be the content, and then from there on, it's all application based. And so if you can just apply a little bit of stuff from the prompt to a graph, you could probably still get a lot of points without actually having covered that content. Plus, you know they're going to pull things that you may have not seen before because they want to see you apply. So even if your teacher hasn't done a certain lab, it's okay. So my students, we've gone through everything, but there's still going to be stuff on that exam that my students still don't know, which is fine. It's a application-based exam, and it's designed to ensure that you understand and you can apply. So. Yeah.
I mean, in a similar vein, like when my students get frustrated that they're getting questions with terminology they've never seen before, or we never learned this, and it's like, you did learn enough to answer this question. Like, trust me a little bit that we would at least give you the foundational skills to apply. And they get there, but a little frustration along the way is healthy.
So are we ready to get it rocking?
Oh, we're ready.
This?
Yep.
Okay. So let's just ease in a little bit first. And someone says, are we going to have to graph on this exam?
My teacher hasn't done a lab all year. Okay. So yes, for paper, no for digital. So I think that's like an unfortunate situation or answer. Regardless, interpreting graphs is a skill you must have. So even if you're not creating graphs from scratch, getting the data, extracting, looking for those overlapping error bars, your appropriate axis scales, knowing your variable groups, those are all things that you'll need regardless. And you can do that without doing labs.
Of course. And those labs are probably going to be labs that you've never seen before anyways. So
yeah, pop over to Bozeman, look at all his labs. He basically has all the videos on all the traditional AP labs.
I actually have my students watch. There's a video that's 1 through 7, and then another one that's 8 through 13. I think each one's like a 10-ish minute video. And my students actually are required to do that for their review notebook that they do. So it's definitely
that's awesome.
Great.
I have one podcast on lab review, but only one at this point.
Well, you just better get on that.
I should do more.
So somebody says, can we go over phases of mitosis and meiosis? So let's just do a generic, what are the phases? Okay. And then we can maybe think about how are they different? Because I think that's like, because if you know all the phases and then you say how they're different to each other, you can apply to either one.
For sure. I mean, so we're going to go for PMAT, right?
Can I do my dance?
Yeah. Oh, all right. Hold on. We had a question. Does anyone have any advice on dealing with self-doubt? Sometimes I get questions wrong because of, the second guessing.
I do that every time.
Practice. The more you go through questions, the stronger your confidence will get. So in that context, it's a little bit of quantity of exposure until you kind of like work it out. It just takes time. Everybody has that. We have that all the time. I'm going to doubt myself 15 times in this session alone.
Oh, it's okay. Her and I do this back and forth. It's great.
All right, let's see the dance. Do it.
Are you ready for my dance? Okay.
I'm ready.
So we have prophase, metaphase, anaphase, telophase,
And then I like to do the cytokinesis too because it's like after it, but remember that's not actually part of mitosis or meiosis. So cytokinesis! So anyways, prophase is when we are preparing to divide. And so you're going to have your chromatin will condense and it will pair up. Metaphase is when whatever it is we're talking about, whether it's homologous chromosomes or sister chromatids, are going to align on the metaphase plate. Anaphase is when whatever it is we're talking about, homologous chromosomes or sister chromatids, they separate, they move apart from each other. And then telophase, or if you're like the person I student taught with, your telophase, as long as you can spell it right, no one knows the difference. So then we have telophase, which is going to be where we have 2 new nuclei. And then, of course, remembering that interphase happens before and cytokinesis.
I always get, so remember, karyokinesis is another term for the division of like the genetic material. So we have kinesis or the snipping or cutting in both cases. My students always get that like moment of like, wait, how many chromosomes are they technically? Like if they're still in the same space, do we have this like pseudo moment of like way too many chromosomes if we're counting? Remember that chromosome numbers is gonna, well, we're gonna get the differences in a second, but when we're talking about mitosis versus meiosis, we're gonna go diploid to diploid in mitosis, whereas we're going diploid to haploid in meiosis, so reducing our chromosome number. Even if our chromosomes are in their duplicate form, right, they're X, they're still connected by a centromere, so we aren't gonna count them as separate until they're pulled apart fully.
And the way that I remember centromere is that centrosome, because a lot of times students get those 2 words confused, is that it's in the middle of my waist. So me, centromere is in my waist. So it's in that actual chromatid middle. I don't know.
I like it.
I don't know. You know we have crazy ways to remember things.
Your teachers are will embarrass themselves to the end of the earth if it helps you to remember something. That I can promise.
Of course.
I don't know what makes.
Did you see me
like,
jumping with the cytokinesis?
When does that happen in teaching? Like that just. I think it's always has to be there. And like the more you teach, the more it becomes evident or comes out. I feel like you have to have it somewhere.
Yeah. Well, you have to be comfortable in your gift and your skill. And the more comfortable we are, the more we just want to show off how awesome it is.
Crazy.
We love our subject. Okay. So somebody has mentioned in the chat, what about prometaphase? And so technically the phases are below level. They're regular level biology, but it's the application of them. And so you have to know those phases in order to apply them to a problem. And so if they were to tell you that, I don't know, there was a cancer drug and the cancer drug is inhibiting the microtubules, then they say, okay, well, where does the cell arrest? So where does the cell get stopped? So you have to think to yourself, okay, well, where's the first time that I see a microtubule actually pulling to do something? And the answer would be that it gets stopped in metaphase. So it gets to metaphase, it's aligned up, but since those microtubules can't depolymerize, they can't shorten, they can't pull it apart, so it's stopped in that metaphase, it doesn't get into anaphase. So it's more of the application that you're going to see on the actual exam.
And being mindful of like the lack of like 3D space, right? So even if the diagram shows it like an up, down, like that's not a thing, right? Where everything's spinning and moving around. So referencing the parts of the cell, these are geographical terms, right? Like towards the poles, that metaphase plate in the equator helps you give directionality to what's happening.
And you can also draw diagrams on the exam. Like if you're looking at a free response, you can draw a diagram and say, as seen in figure 1, label it. What is happening. It definitely label it figure 1 and then refer to it in your text.
Yeah.
And so where do you see the big differences then between mitosis and meiosis then?
A metaphase for the most part, what's lining up, what's pulling apart in anaphase. So if we think about our homologous pairs, right? The homologous pairs do exist anyways in mitosis. They don't go away just because we're not referencing them. So when you talk about what's being separated during metaphase and meiosis, right? We have metaphase 1, where they line up as homologous pairs, and we have metaphase 2, which really just looks like mitosis. And that's it's kind of great that way.
The chromatids.
Right? But if you're trying to distinguish between a cell that might be in metaphase 2 versus one that might be in mitosis, how would you do that?
I would look and see if there's any type of crossover.
Yes, I like that.
Because if there's been crossover occurring, which happened during prophase 1, which is another difference. Prophase 1 is going to show crossing over, which is where we have non-sister chromatids crossing over and exchanging genetic information. This leads to our recombinant chromosomes. Yeah. So when I'm in school, I actually have two students. I have one student that stands next to me, and I cross my leg with the student. Like I kind of like overlap.
You twist it.
Yeah. And then I take off my shoe and they take off their shoe and we exchange shoes.
Do you pick someone with the same shoe size?
Oh, no. It's just fun and games. And they just, it's bad.
That's awesome.
You know, they don't want to take off their shoes. Come on.
One of my actually, I was my interview for my very first teaching job ever, and I did something very similar to that to try and I didn't know the students, I'd never met them, but I was like, let's do an activity.
Yeah. Okay. I think the most awkward thing is when I use cholesterol to symbolize, like in the membrane, I connect it to children in a marriage. That's the most awkward one I use. Anyway, so we already said that crossing over is going to occur in meiosis and does not occur in mitosis. And then we also have independent assortment that occurs in meiosis that does not occur in mitosis. And that independent assortment will occur during metaphase 1. And then another difference is, as you said, what comes apart during anaphase. Mm-hmm. In meiosis, meiosis 1, we're looking at the homologous pairs. And in case you're not sure what that means, that's the chromosome that's the maternal and the paternal chromosome. And when we say maternal and paternal, I'm talking about that when I'm making, like when the egg is being formed in me, right? I'm talking about my mother and my father. Okay. And I think that students sometimes get that a little confused. And then the important thing to note is, when does the cell become haploid in meiosis?
Well, we have to think about what that means, right? So kind of when I went back to counting chromosomes and what do we count as a chromosome, like singular, duplicated. And then when we look at what's dividing, we're actually cutting our homologous pairs in half. So you're going to go to haploid during meiosis 1 from 2N to N. And then in meiosis 2, you're just increasing the quantity of cells that you have by separating your sister chromatids. So that's why meiosis 2 looks so much like mitosis. I always get the question when we talk about crossing over, right? So they're between, when we cross over, it's not between sister chromatids, right? And we think about like why that wouldn't make any sense, right? Sister chromatids are duplicated. They are exactly the same information. So if you crossed over in sister chromatids, you wouldn't be increasing genetic diversity at all.
Sorry.
That's all right.
Okay, so next question that we have. Like, oh my God, there's a lot of questions now in the chat. I like it. So explain mitosis. We said that one. Okay, so you just did a podcast on this. So can you talk about cyclins?
I did. When did I do that?
You wrote me and said, I'm doing something on the CDK. You did probably it was like a month ago. No,
I think it was last year.
Oh, I'm sorry. Well, Kitten, do you want to take this? Do you want me to take it? Okay.
I'm not a huge fan of cyclins and CDKs, quite frankly. So why don't you take this one?
Great, thanks.
You're welcome.
So CDK.
Are you a fan?
No. CDK stands for cyclin-dependent kinase. And a kinase means that it's going to be an enzyme that phosphorylates something. And so cyclin dependent means that it, of course, depends on cyclin. And so the important cyclin dependent kinase that we have is the one that's called MPF. It's the maturation promoting factor. And it's what allows you to get into mitosis and allow for the division. And so what we see is that the CDK level stays at a constant high level. Okay. And then the cyclin level is kind of growing, and kind of we have a higher level as you go through interphase. So as you go through G1, S, G2, and then finally when it reaches that appropriate level, we have this internal signal that now we can go through mitosis. So it's kind of dealing with an internal part to say, okay, yes, we're ready to go. And a student, I have no clue what the username was, corrected me because I actually had a question in the review guide about, where's the connection at? And there's actually CDK that occurs throughout the entire cycle. But the one that we focus on is at MPF. So don't kind of get too overwhelmed. But the big thing is that cyclin is going to be the ligand that binds to it to activate it.
And in the CED, it does state that you don't need to know specific types, more as like the overarching like functionality of them.
Right.
It doesn't mean they won't give them to you.
No,
but
if they give you something that isn't directly stated in the the CED or isn't like a part of our content, they have to give you that background information. So as I said at the beginning, like my students are currently learning about animal systems. If there's an animal system question on the exam and they're wanting you to apply your biology to it, they're going to have to give you the background. So if they give you a nerve cell and they say, okay, you explain how the membrane works, they have to explain to you what an action potential is and how they're sodium potassium. So there's always going to be that background information given to you.
So you mentioned the word, I know you're going to get another question, but while you're
doing
that.
While you're
you
doing that, I'm going to look for a question.
Yeah, I figured. You mentioned the word phosphorylate. It tends to be a scary phrase to students, phosphorylation cascade, right? So we're really referencing a phosphate attaching to typically another protein that changes and modifies the shape of that protein. And I like to think of it like a big domino effect. Phosphorylation tends to activate the pathway, whereas dephosphorylation would deactivate it. Okay, the question's cut off a little bit.
Okay, I know that the question is cut off, but I'm gonna, like, it's the general idea. She's pretty much just giving me the entire CED standard. It says, can we go over the types of inheritance and the different types of dominance? And this is going to lead into the question that we're going to do after this. So I kind of was just doing a quick little thing on heredity. Okay. So the different types of inheritance and dominance. So the big three that we make sure we do know about is complete dominance, which is what we saw with Mendel, right? He had that P and it was an either or trait. Like you either had purple or white flower. So in terms of complete, you have a dominant allele that's able to mask a recessive allele. And so, of course, if there is no dominant allele, then you would see the recessive trait.
Which is why you'll have recessive traits skipping generations in that context. Yeah.
Yeah. And so that's why we'll see that as long as you have one trait, one of the alleles, you can show the dominant. So the homozygous dominant and the heterozygous will look the same. And the FRQ Friday that we did this past Friday actually went into that. It was talking about the active enzyme that was made. And then the next one we have is incomplete dominance. With incomplete dominance, it's like paint mixing, right? So we have one allele that makes red and one allele that makes white. And so if you have both the red and the white allele, you have pink because they just kind of blended in the middle. So the incomplete dominance will be a kind of an in-between or hybrid of the two dominants.
A new phenotypic expression for sure. So that'll be important when you interpret your phenotypic ratios. You're not necessarily going to have 3 to 1. You might have 1 to 2 to 1 or something like that.
Yes. And then with codominance, we're going to see that, again, we have two different dominants, but now you can independently see the two. So in blood types, right, if I have A-type blood and B-type blood, if those two were to mate, I could get an individual that has AB blood. So they have both the A glycolipid and the B glycolipid on their actual cell, and those individual glycolipids are there.
What's interesting about blood type, I love blood type problems. I think they just practice so many skills. We have to remember that even though we have codominance, we also have the recessive allele for O. So you end up with like this multiple alleles scenario. But the pattern of doing these problems is always going to be the same. It comes down to your interpretation of the results. And I think that that's kind of where your analysis comes in terms of explaining your ideas. Being able to do the Punnett square is really just like a super small piece of the puzzle, because if you don't actually read the prompt and read what the question is asking, you could interpret it incorrectly because you don't actually answer the question, right? You get so excited that you got an answer, but is it the answer they're looking for, right?
And speaking about those ratios, if you were to see a free response question, please make sure you write in a complete sentence. Don't just write out the ratio. You need to say the ratio is, and then give out that ratio. And if they say like phenotypic ratio, give out those phenotypes. 1 red, 2 pink, 1 white. And you have to give it out with the actual phenotypes included.
Do they don't have a preference between fractions or like colons, do they?
So there's a slight thing.
Let's hear it.
As long
just
as
to be clear,
Tiffany's
has graded. I have not graded. So Tiffany has the insight on the grading.
I've been grading this. I think it's been 6 years, 5 years. I really don't remember how many years it's been. At that
Too
point,
many.
And so as long as the ratio matches, okay. So for example, like a 1 to 1 to 1 to 1 ratio means you have a fourth of each of them. So as long as you wrote down, 1 to 1 to 1, or a fourth to fourth to fourth to fourth, 4, 4, 4, 4, 4, it doesn't matter. The problem comes into play if you're trying to write that a 1/2, right? You write 1.2. That doesn't mean 1/2. That means 1/3 and 2/3. And that's where students have issues, is they don't understand the way the ratios work in terms of the semicolons. So you always just write out pretending like you have the same denominator for all of them. That's
great. That was good feedback. So the way that you practice now, just be consistent, because you don't want to cut corners now and then make those errors later.
And that's why whenever you write your responses, always write complete sentences. Even now, when you're practicing, just write your complete sentences out.
Yep.
Okay, so polygenic, we've got, okay, so we did everything there. In terms of linkage, though, there's two different types, right? You have autosomal and a linked trait. What's the difference there?
So if we're talking about autosomal, we have to be mindful of like which chromosomes we're referencing. So autosomal is going to refer to something on chromosomes 1 to 22. What was the second one you said? A linked trait?
Yeah, like let's say sex linked.
Oh, okay. Right. So okay. So it's associated.
Coming
right.
Next. You know what's coming next. Come on.
All right. So associated with chromosome 23, right? So chromosome 23 has a unique inheritance pattern, and so if we're doing Punnett squares that are sex-linked traits, we can't just include like our regular capital letters, but we have to associate them to the X or the Y. Most, but not all, most sex-linked traits are going to be on the X chromosome, and they are often recessive. However, that is not an absolute with that. So if we're talking sex-linked recessive disorders, I think that's what the next question is going to be. Trying to keep
because of pedigrees.
Oh. Well, that's okay. How do you know if it's autosomal or sex-linked, dominant or recessive? Yeah.
So you kind of want to follow the pathway for a second, right? So you need to look at what you have in your pedigree. And so if it's dominant, you will see it in every generation. There's no skipping of a generation. So if you have unaffected parents, there's no way you will get an affected child if it's a dominant trait. So you're first looking at that, looking at dominant versus recessive. And then after that, you look for the sex-linked versus the autosomal. Autosomal, you have an equal chance for males and females, versus sex-linked, there's going to be a higher chance of males having it. And this is when we think about it being an X-linked trait, because of the fact that males only have one X chromosome. So you either have the X that has the trait on it, or you don't have the X with the trait on it. So you
can't cover it up at all. There's nothing to hide it.
Whereas the female, if we're looking at an X-linked recessive, there's a chance that she could have gotten a X that was dominant that could mask it. And so if you're looking for it, you'll find that there is going to be a affected male that had a mother with the trait, or a mother that was a carrier of it. So you always see the affected male coming from an affected female, or a carrier female. You have to follow the path.
And like, it's a skill. I say the more you look at, the more you kind of pick up on it faster. But if you're kind of backed into a corner and you aren't quite sure, pick something and try it, right? And pedigrees aren't going to prove that it's a particular type of inheritance, but it can disprove others.
And someone in the chat reminded us that we didn't talk about polygenic.
Poly meaning many traits, many different chromosomal components affecting one trait. Eye color is polygenic.
Skin color, height, all of those.
Anything
you see on
humans
a spectrum.
Don't follow a ton of like normal inheritance patterns. I mean, in a lot of ways, Mendel kind of lucked out with his P's.
He really did luck out.
He really did.
He really did. But yeah, so anything that you see that's on a spectrum, that's where you're usually going to see polygenic.
Yeah.
So this is Unit 4 and 5. It's kind of a blend of different things. Just because
Unit 4 is going to be cell communication and cell cycle. Unit 5 is going to cover heredity. So we're kind of like in both spheres right now.
Okay, so let's just jump back over to 4, because the people are wanting some more 4 stuff.
Does he want checkpoints? Is that what he wants?
Yeah, they want to know about the regulation. So
I like G-Naught. G-Naught's one of my faves.
Okay, what's G-Naughts?
G-Naught, G sub 0.
Right?
Oh, G-Naught. Sorry. Yeah, G0. G sub.
I thought you were giving me an acronym or some like mnemonic device. I'm like, yes.
No.
Okay.
Right, the cells that don't divide.
Okay.
The cells that kind of buck the system, right?
Awesome.
They leave. They never go into S phase, or at least not until signaled. Yeah.
Okay.
Fun.
So that G1 checkpoint is where we get to G0, or G0 as I call it. And that has to do with, do you get the go-ahead signal, right? Do you have that growth factor that allows you to move past this phase? If you can move past G1, you're probably going to divide. What's our next checkpoint?
After S phase, so you want to look at your spindle fibers were formed.
All the DNA is divided.
Yeah, all your DNA is made.
Not divided, multiplied.
Not divided, multiplied. You have all your copies. Yeah.
And then we have our M checkpoint, which is my favorite checkpoint.
Okay.
And it has to do with, are all of the kinetochore microtubules attached to the centromeres of your chromatids? Because if it's not, that's what leads you into non-disjunction, because of the fact
we didn't talk about non-disjunction.
No, we didn't. That's why I'm leaving it there now.
There you go.
And so with non-disjunction, that involves trisomy 21, in which you can get an extra copy of a chromosome because of the fact that there wasn't a kinetochore microtubule or a microtubule attached to both poles attached to the two different chromatids to equally separate them. And so if they're not all attached, then of course you move in. Now, we're giving you a rough what the checkpoints are. On the exam, they're probably going to give you some information. So for example, they may tell you that in the M checkpoint, it activates this enzyme called separase, and separase does, and they go through the whole process that it's going to separate the two chromatids, and then they may tell you that there's, I don't know, some inhibitor that inhibits separase. What would be the product? And they may ask you an application from there.
And since we're talking about kind of like the cell cycle and the going through the cell cycle, it is worth noting that there is no interphase in between meiosis 1 and meiosis 2.
It's called something. Is it like interkinesis?
Something like that.
Inter something.
It's very brief.
I mean,
well, it depends on the cell, right? The type of cell that we're talking about in terms of the timing of that. It's not going to be the same for all types of gametes. You know, not that you need to know the depth of this within specific species, but it could be part of examples. So like male spermatogenesis is going to complete meiosis 2. Whereas for females, I'm talking about humans specifically right now, you're not going to have meiosis 2 unless there's actually fertilization. So there's like an arrest in meiosis 1 where the cell doesn't finish dividing until fertilization occurs.
I like to say that word. Ooh, genesis. So much fun.
It is fun to say. Two consecutive O's. Ooh, genesis.
Ooh, genesis. I'm sorry. I should be looking at questions.
No, that's all right. We get distracted by ourselves and our terminology.
So how about you tell me about this digital exam while I find some more questions?
Sure. So the digital exam is so we're specifically talking about biology. So for biology, you will have a digital choice and only digital choice for the second and the third test dates. The digital exam can be taken at school or at home, and it's going to be uniquely designed. So first of all, you have to download the specific College Board application. You have to use it a couple days in advance to like kind of go through your security pieces. And then once you're in it, technically, you don't need to have Wi-Fi the whole time. It could cut out, but you will need to have all of your responses typed into it. So if you took AP exams last year, you're not going to be able to like handwrite anything. There's not going to be any uploads or submissions for that. The key thing with the digital exam is there's no going back. You have to go forward. So for our friend that asked about how do you not double guess yourself, there's no room for that. You can't go back and check your work. You cannot go back to a previous question. So timing, being able to time yourself and keep your own pacing is huge for both exams, but for the digital exam in particular. And if you choose to take, or if your school chooses to have you take the AP bio exam digitally, that does not mean that you have to take all of your AP exams digitally. Each of them are distinctive, but it is a school decision. So if you aren't sure what's happening for your AP exam yet, talk to your school's coordinator.
Okay, so our next question is, what's the difference between chromatin and chromatids?
Isn't it a curse that everything has to begin with the same first 4 to 5 letters?
I don't know. I think they just want to confuse us. Like,
give us
all these
it's
words that are very
intentional.
similar. Like, come on.
Right. So I always call chromatin the spaghetti, the spaghetti version, and then chromatids. You start to coil up your spaghetti on your fork. It's going to get more dense, right? So I'm always hungry. I think my food analogies are constant.
Chromatin analogy.
Do we? Oh, love it. Chromatin. Okay, so to me, it's what is it? Not only what is it, but when do you see it, right? So chromatin is loose DNA, not coiled and condensed. It doesn't mean it's not wrapped around histones, because it still is. That histone is a part of gene regulation. So which parts of DNA are tightly wrapped around histones or not? Histones are proteins. Everything's practically proteins. And then your chromatid is going to be 1/2 of your duplicated sister match here. So when you're totally coiled, and you'll see those chromatids, they become denser. That's when you start to be able to see DNA under the microscope. Yeah.
And one thing I've seen, like, is why does the DNA wrap around that histone? Like, what functionality or what is the polarity of these two components in order to allow the DNA to wrap around the histone? And so you kind of have to think about, well, DNA is made up of, sugar and phosphate has a 2- charge to it. So DNA is negatively charged. And then your histone is positively charged because of the lysine tail. There's these little like lysine tails that stick out. And so the lysine tail interacts with the DNA and it wraps it around like spaghetti around a fork.
Yeah, I used an analogy the other day about like, those, well, now it doesn't work, right? So we're all using wireless headphones. So it's those little packs we used to have to wrap our headphone cords around to keep them less tangled. But the analogy doesn't even work anymore.
Well, the students showed me this cool way you could use your fingers like to wrap it around, and then like it makes it like a nice little
like a bow.
Yeah. They showed me a couple years ago. I was like, oh, this is amazing. I was very happy about that one. Sorry. Okay, so I don't know if feedback is in this unit. I think it is. Is feedback in this unit? Okay, let's do what's the difference between positive and negative feedback?
Homeostasis. Sorry, I'm responding to a question in the feed.
Okay, so I always think about negative feedback as bringing you back to normal, versus positive feedback is going to be more of a buildup. And so like if we think about like, I don't know, insulin, right? When you just ate your dinner, right? You have all this sugar in your blood sugar is too high, right? So insulin is going to be released to bring your blood sugar level back down to a normal level. And if you happen to, you're fasting for some reason and your blood glucose level is too low, then your body's going to release something called glucagon, when the glucose is gone, to help you to bring the glucose level back up, because it causes your liver to release the glycogen, breaks down the glycogen into glucose to release the glucose into your blood to bring the blood glucose level back up. So that's all negative feedback. We're bringing ourselves back to normal.
And negative feedback is more common. That is the more common example. Because homeostasis is a condition of being living, right? And the ability to maintain your constant internal conditions despite changes.
And then positive feedback is going to kind of, in my eyes, building up to some type of response. So I gave my students the example of oxytocin and how oxytocin works with childbirth. And we're not going to get into all the details, but overall, the oxytocin causes more contractions, which ends up leading to the birth of the child.
Right. The other example that works for positive feedback is with fruit ripening. So you have ethylene being produced. Yeah, ethylene being produced. Right. There's all those like old wives' tricks of like how to ripen your avocado faster, but there's a lot of truth to it. Specifically chemical science. So the concept of like one bad apple can spoil the bunch, like that's really true. We think about why this would be an advantage, right? We go into like evolution and ecology kind of like going forward. Like why would you want all the fruits to ripen simultaneously? What would be an advantage to that? And if you think about
together and distribute the seeds.
Right. So if we're going to bring over some sort of animal to help with the process of distributing seeds throughout the land, why not have them do it all at once and a little gorge fest. So yeah,
that's pretty cool. I also like the way ethylene works, because it also has that triple like effect where like if there's like a rock above it, it can grow horizontal around the rock. I always love that about ethylene. Cool.
I like the like anything zombie apocalypse, nature will find a way. Nature will find a way.
Okay. Cancer's in this unit, right?
Yeah, general things about like stuff going wonky.
Oncogenes and proto-oncogenes?
Apoptosis is in this unit. I don't think it is. Is it oncogenes?
Well, let's just go ahead and mention it because I already have it on the screen. Sorry.
Okay, that's all right.
Oncogenes are going to be your cancer-causing genes. We also connected back to units before, I promise. Oncogenes and proto-oncogenes. So proto-oncogenes are your normal cell division genes. And so if they've been mutated and cause a excess of cell division, they are converted from proto-oncogenes into an oncogene. See, it connects.
It connects. So really what you're going to end up with is too many cells in one space. They're crowded. Normally we have some sort of like density inhibition that can occur. I mean, there's a lot of things that can be, we didn't really talk about like cell communication much yet, but there are a couple of factors here that can influence when cells can figure out what to do, what's adjacent to it.
And then let's talk about cell communication then. Because the question's in there. Yes, P53, the angel of death. That P53 is involved in everything if we're talking about cells in cancer. So let's talk about the transduction pathway. So what's our first step of transduction, or first cell signaling?
Well, how far back you want to go?
Reception,
transduction, and response?
Yes, let's go all the way through the whole process and thinking about the different types of ligands we could have between polar and non-polar.
Right, ligands. Where it's going to bind. Okay.
So, am I starting? You started?
You start. I'll pick up on the ligand.
Sounds great. So if we're talking about a non-polar ligand, the non-polar ligand, since it's non-polar, can pass through the plasma membrane, and it will go and find a receptor that's intracellular. So the receptor will be on the inside of the cell to bind. Versus if we're looking at a polar ligand, then it's going to bind on the membrane because the fact that polar can't get through the membrane. So we often talk about hormones in this aspect. So your polar would be your protein hormones, and your non-polar would be like your steroid hormones, which connects you back to unit 1.
Right, and your lipids. Yeah, so depending upon where that ligand binds, that's going to be leading into our transduction, like the actual middle part of the pathway. And this goes back to what I said about phosphorylation cascades and what's the purpose of this as this domino effect of phosphorylation. It's to amplify the signal. What the signal is really depends upon what's being communicated. But you are inside the cell at this point, somewhere in the cytoplasm, typically. And once you have this phosphorylation cascade where you are going to have protein after protein kind of like relaying the signal along, you're going to get towards your response. Let's say the response is going to happen inside the nucleus. What might that response be?
It can act as a transcription factor to turn on or turn off genes. And so the transcription factor is going to cause the RNA polymerase to be more likely to bind or less likely to bind. And so that can lead to us making a protein or not making a protein. It could also lead to apoptosis, so making a protein that causes the cell to digest from the inside out, could cause cell growth. Cause the cell to grow in a certain way. That's what we see with the yeast, with the A factor and the alpha factor. They grow towards each other so they can mate.
I mean, right now, everybody's worried about their antibodies being produced for certain reasons that might be happening in our environment. So a really good example of response, right? So if we talk about, B cell, which is going to be an extracellular receptor binding to an example, in this case, it could be the COVID spike, right? Onto that B cell as the initiation for, okay, cascade, go make me an antibody for this particular spike protein.
And then the antibody then becomes a signal for the macrophages to engulf it to help us to get rid of it a little bit more. So there's all this stuff. Something that we do want to also bring in terms of that cascade, it doesn't always have to be a phosphorylation cascade. Sometimes it can be a secondary messenger. Calcium is a secondary messenger. You see that in your muscle cells, like the calcium is held in the smoothie R, which in the muscle cells called the sarcoplasmic
reticulum. Reticulum means net, remember? So it's going to look like a net on the cell.
And do you know that's why they call it reticulum? Because that's what the old purses were called. They were reticula, and they just look like big nets. Cool fun fact, sorry. But so the calcium binds to a component that allows for basically the rearrangement of the actin and myosin so they can bind together.
Cyclic AMP, right? So cyclic adenosine monophosphate.
Right. My students had the hardest time understanding what cyclic meant. So there's actually it's a ring structure on it.
Yeah.
Yes. There's a ring. It's cyclic, and then adenosine monophosphate. There's only one. And so it's another signal that works. I think that one works with the
G-coupled protein receptor, right?
Yeah, and you see how we're kind of fumbling over this?
Yeah. I'm trying to see it in my head.
You're good. FYI, on the exam, they're going to give you a pathway. They're going to show you the full pathway, and they're going to ask you an application about that pathway. We just did a free response one on the immune system, and it was like the gastrodermin and then the interleukin being released, and they showed you the pathway about what was happening, and you just had to explain, if this didn't happen, then this would or wouldn't happen. So you don't have to have all these pathways memorized. You have to have a general idea of what does reception mean, what does transduction mean, and then what are different responses that could occur. But you don't have to be able to know, well, insulin is a G protein coupled receptor, and then insulin causes You don't need to know all that detail because it's different for every single cell. If it has a different receptor but still binds to the same signal, then it could have a different response.
Yep.
If there's different relay molecules, it could have a different response. So epinephrine is a great example of this, right? Epinephrine is going to cause vasodilation in blood vessels going to your muscles, but it causes vasoconstriction to blood vessels going to your GI tract. And then it's going to cause the cells of your liver to release glucose. So there's 3 different responses from the same signal. So it's very hard for you to predict what's going to happen.
And I think that's what's interesting about us having these lives is like in our heads, we're trying to talk like broadly about processes when in reality, like the way we teach it is through example. So you're kind of like flip-flopping. One of my students, we did cell communication. We did the stop motion project, and he wanted to do interleukins, and I just like I let him try for like a day, and he just was like it was too complicated.
Oh yeah, interleukin's really cool. They were going to give my mom that back in the day when she was going through her treatments for things. Wow. Okay, so I guess we can jump back into inheritance. I'm sorry.
No, we just have one question above. It was above cyclic AMP. I think you just talked about this in stories the other day. How can a signal change the phenotype of an organism? I think he's referring to phenotypic plasticity.
Yeah, so it could cause the production of certain genes. Like I guess the best example I can come up with on the top of my head right now is the morning glory. Morning glories are going to be blue, and then at certain times they become pink. I could have that backwards.
Sounds correct.
But there was a first response question a couple of years ago, and it had to do with the pH of the soil, I believe. And so as the pH changes, it's then going to lead to a change within the cell. And I believe it caused water to flow into an area. When the water moved into the area, then it caused the change in the color.
Isn't that true for hydrangeas as well? The pH of the soil influences. Yeah.
So again, you're not gonna have to know which cells do this. You're just gonna have to be able to understand what would be the purpose of it. So an evolutionary purpose would be the snowshoe hare can be white during the winter months, so it blends into its environment, and then it is able to produce a different protein during the summer months to have a brown coat. So again, camouflage into the environment, and it isn't going to be more likely to eat. And
so that's the same genotype, right? Genotype is not changing in this example. It's just the expression of that genotype.
And when we talk about acidity, we're kind of thinking about that the more acidic something is, the higher concentration of protons and the lower the number. Like if it's acidic, it's in that negative, it's like 0 to 7. And then alkaline or basic is above 7 to 14. And you see I'm putting these in quotes because if you know anything about chemistry, that's not the scale we're on. We go farther than that, but we're just going to stick within 0 to 14 because that's where most of life is.
And it is logarithmic, so it's a 10-fold change as you go up the pH scale. You will not have to calculate pH, interpret pH, and the pH equation is on your equation sheet, just in case you want to look at it.
And so I always tell them to look at the exponent, right? Because if we're looking at a 10 to the 4 compared to 10 to the 6, it's 100 times difference. You just kind of use your exponent rules that you learned in algebra. Yes. Okay, so let's jump back into heredity for just a moment. Thinking about non-nuclear inheritance. And I believe you did something on this recently with your podcast.
Is that what the podcast is? Yeah, it's a non-nuclear inheritance. So if you're not getting it from your nucleus, where is it coming from? So love pedigrees with mitochondrial inheritance. I think that they're super cool to interpret. Do you have one like on hand? Curious.
Awesome. I do not have it in my. I was not prepared for this. Right. I'm very sorry.
So just because in the CED, it does state that mitochondrial inheritance is maternal. So that's going to be a mother.
And chloroplast.
And chloroplast. And chloroplast, right? And that's going to have to do with back to the endosymbiosis theory, the fact that they were once independent prokaryotes and they have their own DNA. And so you can have the expression of those particular genes. So if you're looking for it on a pedigree, you are going to want to follow that maternal lineage inheritance pattern.
All of the offspring from the mother will be affected. And so if you see an affected mother and all of her offspring, then you could, connect that and say, okay, well, this is a non-nuclear inheritance. It's mitochondrial or this is from the chloroplast.
In your scrubbing of old questions, have you seen it?
The mitochondria? Yeah.
Mm-hmm.
Oh, yeah. It's been in the old tests. They pick unique situations. So as you were saying earlier about, well, they don't usually do Y-links. Well, you're right. Y-links is a rare thing. And X-linked dominant is also a rare thing. Which tends to mean that it might show up. I've never seen a Y-linked on the exam. I've never seen an X-linked dominant that I can remember.
Right.
But just because it's never happened before doesn't mean it won't happen now. I always like to be prepared for everything. I'm always scared. Okay, so let's see. Oh my gosh, I can't.
While you're doing that, I'm going to pan through the CED just to see what we skipped, if we want to come back and cover anything.
We did that. Okay, so I guess we can talk about the math for a second then, while she's panning through the CED. How do we know when to do a dihybrid cross or a regular Punnett square? Okay, so I'm going to tell you my personal opinion. And I'm just one teacher, and
you like to do them separate, don't you?
I do like to do them separate. My belief is that it's a lot easier to do a 2x2 square and another 2x2 square, and then multiply the ratios. So if they're asking you how many individuals, I don't know, yellow round, and you do this big 4x4. Number one, you had to fill in the 4x4 square. Then you had to look through each individual one and say, okay, well, yellow round, yellow, oh no, and like it takes a lot of time to sit there looking through your Punnett square, right? Versus if you go and do let's pretend we're looking at a dihybrid cross, right? So we do heterozygous for yellow. So we say, okay, well, 3/4 are yellow. Heterozygous for round, 3/4 are round. Then I can immediately say, okay, well, 3/4 times 3/4, 9/16, boom. And I have my answer versus having to do this big square and then searching for the things. Like we're talking about, you only have about a minute to a minute and a half on the AP exam for each of your multiple choice questions. And depending on which question we're looking at, because they can give you a chi-square question in a multiple choice question, okay, in which you have to be able to go through the chi-square calculation, which means you need to be able to quickly know your ratios and be able to do your whole chi-square calculation. And speaking of which, some of your questions are probably going to take longer than a minute and a half, and some of them will take less than a minute and a half. And so there's that balance that's going to happen with your electronic exam, that you need to make sure that you at least have, I guess, a minute and a half. Like you
on
can't
average,
spend more than a minute and a half on the question. Like if you find that you're getting close to that time, you got to pick one and move on. And maybe talk to your math teacher and say, okay, is there a quick way that I could look at these problems and say, okay, around where will my number be? And maybe that can help you to eliminate some of your answer choices if you run out of time and can't go through the full chi-square calculation.
So for digital test takers, make sure you have some scratch paper nearby. 100%, just because you can't, and this might be a good practice. The best way to practice to date, the best way to practice your digital exam is to do AP Classroom, because that is digital, right?
And don't press back.
And don't press back, right? So you have to create the circumstances by which you will be testing. So if you're going to wear a watch, if you're going to have your paper off to the right, I know this sounds really like archaic, but so I've taught I went to and I've taught in schools where all the students wear uniforms, and there's a lot of educational data behind consistency and how you study, how you test, and how you learn. So it's about associative education, basically. So the way that you practice will influence the way you perform. So you want to create that as much as possible. All my students always ask, can we have a dress down day for exams? And it's like, that's like the opposite of what will help you do well on exams, even though sweatpants are lovely.
Oh yeah, totally lovely. I was thinking about something as you were talking about, I've forgotten it. So also something for your digital exam, maybe ask your teacher if you can have a bubble sheet. And so when you're going through and saying, okay, well, I know the answer is not C, not D, and you can kind of cross things out on your bubble sheet, because you won't be able to manipulate the screen, right? So it's a way that you can cross answers off without actually being able to cross them off.
And FRQ annotation is going to look a little different. So shorthand things that you're going to be able to do in order to kind of break down the questions where you can't underline itself. Coming up with that pattern now will be a good idea, even if it's just like summarizing the graph or chart in a sentence so that you can extract the most important information and have that to work with would be really good.
Okay, so we have one more question, and then I think that we need to close it out. And then I'll also answer the question that Samantha just asked in a second. I'll answer Samantha's question in just one moment. So it says, is a chromosome a pair of sister chromatids? Okay.
Can be.
It can be. A chromosome could either be the pair of sister chromatids, or once they separate, now they're called chromosomes. And so it all depends on where you are in the cell cycle, on what you're going to call them and how you're going to address them. But yeah, a pair is technically a chromosome. And so Samantha wants to know, is there any released multiple choice exams from College Board? The only released exam that you have access to as a student is the 2013 exam. So if you just type into Google 2013 AP Bio exam, it'll pop up. It's on the College Board website, easy peasy, lemon squeezy. And yours right there. If you want to have an interactive way that will, grade it for you, I just opened up a quizzes game that has the 2013 exam on it. Your teachers have 3 practice exams. So if you're a virtual student, then as long as you do AP Classroom during school time with them, then you can use the lockdown browser and you can take a practice exam. If you're in person, talk to your teacher about saying, hey, can we do a stay after school for a 3-hour span? Can we do a Saturday session in which you can sit and take that practice exam? Because I don't know about y'all, but my exams for my students are not 3 hours long, and they're not used to taking a 3-hour exam, especially now that we've been virtual for so long. Like, they're not even used to sitting in a chair with pants on. They're not used to like wearing like appropriate for school pants.
Right. They don't have the stamina that they used to.
Right.
And teachers need either. Like, there's a different stamina we have to readjust to as well.
So I would talk to them like, my students are virtual, but they're all going to come to school, masks on, 6 feet apart, and we're going to sit for a 3-hour exam. They'll take their multiple choice, and then they're going to have like a 5-minute break, and then they'll do their free response. They will hand write their free responses because I'm motivating them to do the paper exam. And so I'm stimulating those conditions. And I'm going to work on making an Excel file for you students that will allow you that once you do the exam with your teacher, you can enter your multiple choice answers, and it will tell you whether you which areas you need to work on. So it'll break it down by the CED and tell you which units you're weak in. It'll tell you which big ideas you're weak in, and kind of give you a rough sketch of your understanding, which I think is great.
And because that's essential. Like, during April, you got to start to narrow down in what do you need to review and what can you just let go? Because there's definitely pieces of the exam that that you need to trust that If you know it today, you'll know it in May. And so being able to kind of compartmentalize your studying moving forward is a skill. And it's also like, oh, thank goodness. Like, it's a relief, right? So if out of the 8 units, 4 of them you feel awesome in, then like put them off to the side for a while and then focus on the 4 you need to.
Yeah. And a student asked me today, he's like, well, how do I study for this? I said, well, take a practice exam. Find where your weaknesses are and focus on your weaknesses, because your strengths are going to, of course, stretch, right?
So I don't know about you, but I give my students an FRQ on the very first day of school. They've never seen anything for AP Bio ever. And I've just held on to it. It's been in my desk drawer this whole time. And the moment we're done with Unit 8, I'm going to give it to them again.
So my students, and I don't know how many of them are currently on this call, but my students do the exact same full-length AP exam on the first day of school.
Oh, you got a full
their
test?
First semester final exam, and as their second semester final exam. It's the exact same test, and I can tell them how they do over the whole sem. I can tell them based on the first semester, hey, this is potential score you're going to make, in May. I have all this data analysis that I do from it. They've seen this exam so many times, but yet they don't remember that they've seen it until they sit down to take it. They're like, we've seen this question before. Yeah, I know. You'll figure it out. It's cool. Sorry. So it is now 9:00. Last minute tips before we head out.
Deep breath.
Deep breaths.
Deep breaths.
And so this is Melanie. She's from the APsolute RecAP. So if you're looking for a podcast that you can listen to, 10 minutes, pop it in your ear, go for a walk and get some blood flowing. And it's a great way to review. And then I'm Tiffany Jones from AP Penguins, currently doing daily review on the Instagram. And there's 20 million other things that I'm doing, like FRQ Fridays, these lives with Melanie.
And whatever you ask her to do in the next few weeks, she might say yes to.
And I also just this morning opened up, oh my God, I'm going to assume it's probably like 30 quizzes, that website game. And so if you go to the website, you can pull all of those codes and they're all open and available for you to practice with. So,
and I think that there's one thing to be said. There are resources if you look. I think from a student perspective, you just have to start to like take it in, because no matter how many resources you're exposed to, you still have to use them properly for them to be effective.
What's that quote from Harry Potter? Something about the help will always be given at Hogwarts. Oh, you know to turn on the light or something. What is that?
Yeah, quote? For those who ask for it or something.
Oh, now I wish I knew that quote.
Dumbledore's a genius, and that's all we need to know.
Of course.
Right.
So last thing, you are all AP Bio Penguins, and you are all dressed for success. Have a
great night, everybody. Bye.
Bye.